Hello everyone, my name is Caitlyn Fourie. I am at the University of Pretoria in South Africa at the Center for Microbial Ecology and Genomics. I'm a first year master's student. I am currently doing my project titled Sub-Antarctic Indian Ocean Microbial Gene Duplications, which is supervised by Pratulani Makalanyane. Gene duplications are a common evolutionary response to the changing selection pressures in bacteria that are a result of the environment in which they live changing, specifically when altering their gene expression is not sufficient to overcome the changing conditions. Gene duplications that have diverged and developed novel functions allow an organism to adapt to its environment by expressing a new phenotype. It has been found to be a great source of genetic innovation. The extent of gene duplications and its contribution towards adaptation has been studied in many different bacteria and other organisms, while the rates of gene duplication were suggested to be very high. The extent of microbial gene duplications in the subantarctic Indian Ocean at Marion Island and between Marion Island and South Africa is largely understudied. There are also few metagenomic studies that have been conducted in this area, so there is a need for more data. This study will aid us in our understanding of the extent of gene duplications in the subantarctic Indian Ocean microbial communities, as well as their role in the adaptation of these microbes to the extreme environments they face here. I hypothesize that microbial species will contain significant duplications that have diverged to develop new functions that allow the species to adapt to the environment here, since gene duplications act as a source of genetic innovation. So my master's project is split into two sections. The first, I'll be looking at gene duplications in single-cell assembled genomes that we already have at CMEG. And then the second part of my project, I will be looking at metagenome assembled genomes that we have created during this year from Marion Island. So to look at gene duplications in the SAGs that we have at CMEG, I'm going to use a MinION device to resequence the single-cell genomes in order to complete the genomes that we have. These reads will then be used to look for gene duplications in each of the SAGs using a BLAST-P all-against-all search. And then I will finally conduct a genus-level pangenomic analysis of important SAGs by downloading genomes of each genera from the NCBI, and I'll use Fourie to calculate the pangenome with a 75% sequence identity. So for the second part of my project, where I look at gene duplications and metagenome assembled genomes, or MAGs, we collected seawater between South Africa and Marion Island, which is in between South Africa and Antarctica, and as well as around Marion Island. We then filtered this water using polycarbonate filters to collect the microbes on these filters, and then we extracted their DNA and sent the DNA for shotgun metagenomic sequencing in the United States. And we are currently assembling the MAGs from these reads. And then once they are assembled, I'll do a gene duplication analysis using the same methods that I used for the gene duplications in the SAGs. And then once that's done, I will use nutrient analysis and enzyme activity assays to correlate with this data and see which paralogs are necessary for survival of these species. So that's my project in a nutshell, and I'd just like to thank AtlantECO, the University of Pretoria, and CMEG for their contributions. Thank you.